This is part 2 of our topic on microbial infection. We're going to continue talking about pneumonia through this slide set, but we're now going to look at the things other than bacteria that can be a cause for pneumonia. We'll also have a look at the drugs that are used for those other microbes. We're going to have a look at the structure of viruses and the differences that we see in protozoans and fungi and parasites and how they're different in their structure compared to viruses and bacteria. We'll also look at pneumonia caused by these viruses, protozoans, fungi and parasites. And then we'll have a look at the antiviral drugs, the antiprotozoal drugs, the antifungal drugs and the antiparasitic drugs. And so we'll be looking at the drug groups, but we'll give an example in each case as to how it's used in pneumonia. So let's start with the viruses. Respiratory syncytial virus, RSV virus, is one of the major causes for pneumonia in children and adults, but particularly in children and particularly those with heart and lung disease. So 2,500 to 4,500 hospitalisations a year are courtesy of respiratory syncytial virus, or RSV. Now, this viral surface has spikes on it that fuse the infested host cells together, and the word syncytial comes from this structure that is formed when these cells fuse together called a syncytia. And so syncytia means multinucleated mass of cells. So the virus gets into the cells then start to fuse together, those that have been commonly infected by the virus, and they form a clump, a mass. Other viruses that are known to cause pneumonia are the influenza virus, adenoviruses, and one that came up in 2004 and that had the world rather worried was severe acute respiratory syndrome, and that's SARS. And so it's a coronavirus that originates in animals, and it's thought to cross to humans in crowded environments. So this spread through Asia, and courtesy of our world shrinking with air traffic and planes going around the world, started to spread out from there. So spread by respiratory droplets and contaminated hands or objects. And so they were very much worried about SARS becoming a global epidemic at the time that the last outbreak occurred. So the thing about viral pneumonia is that it's usually self-limiting, and by that we mean it will run its course regardless of anything else you do. And if anything, if you turn around and allow yourself to be sick and let the virus do its thing, you are likely to turn around and get over it faster, because your immune system can spend time worrying about that and dealing with that virus than you turning around and stressing your body system on multiple fronts at the same time. The thing about the viral pneumonias is that the virus can damage the ciliated epithelial cells, which we noted in part 1, and because of that, that means our mucociliary escalator is not going to move the mucus up and out of the lungs as easily, and that means bacteria can get into the lungs and it will stay there and become established. So a viral infection in the lungs can lead to a secondary bacterial infection in the lungs. Now the virus also invades the goblet cells. The goblet cells are the cells of the bronchial tract that are making mucus, and as it turns around and uses that cell's machinery to make new virus particles, it can cause the death of those cells, so they slough off, they fall off the bronchial walls. And the inflammatory response that is the result of that makes those bronchial walls quite edematous, and so that narrows down the airway passages, making it harder to breathe. And so the x-ray there shows you an x-ray of somebody who has had pneumonia with influenza virus as the cause, and you can see that it's very multifocal, unlike the x-rays we looked at in part 1, and the consolidations are quite patchy. So let's talk about the structure of a virus. And you can see in the image that's labelled B, they're micrographs of virus particles. You certainly can't see them underneath a microscope because they're far too small. So you need an electron microscope to actually see them. And you can see that the outside coating on number 1 and number 3 is kind of spiky. And in fact, number 1 looks a bit like a wattle flower. And so even if you look at number 2, if you look at it really closely, you can see that there's these sort of projections coming off the outside edge, and that it's got this multi-layered sort of circular structure within it. And again, you can see that in number 4 as well. So number 1 is in fact an adenovirus. Number 2 is the Epstein-Barr virus, which is the cause of glandular fever. Number 3 is rotavirus, and number 4 is a paramyxovirus. And so when we draw those diagrammatically, we get the diagram that we see on the left. And so in diagram A, we can see this outside coating that we call a capsid, and within it is either DNA or RNA. And if that naked virus has an envelope around it, then we see the diagram below it where it's actually got this double layer over the top of this capsid. And the double layer, there's a lipid bilayer as well as an internal protein coat inside that. And then we have these spikes coming off it, which are the glycoprotein spikes. Now, some viruses, rather than having a capsid, the DNA within the glycoprotein and lipid coating is just RNA. So you can see there that the structures are slightly different depending on the strain of virus, but there are elements to it that can be common across viruses. So, but they either have a DNA or an RNA. They never have both in the center, and then they may have variations of numbers of layers around that DNA or RNA. And those spikes and those layers enable it to attach to a host cell. So in our flowchart on the right, where we have the cell itself, the host cell being the big blue squarey thing, and we have our virus attaching to the receptor in the cell membrane. And viruses are actually quite selective for the cells they invade. So the virus finds a receptor that it's appropriate for, attaches to it, moves inside the cell, and then uncoats those protective layers. And that then exposes the viral DNA or RNA, and that then has to become part of the cellular DNA. So we get viral genome replication and we get mRNA synthesis, making more viral proteins. And once we have more viral proteins, it will also create any other viral inclusions that need to be there. It will create the capsids and things that need to be around the virus in order, when it's ejected, it looks like the parent virus. And in order to do that, it will cause some metabolic derangements. It might cause cell lysis in order to release all those new viruses. It's possible that that interaction with the DNA can cause neoplasia. And of course, because it's foreign to the system, and if there are viral antigens produced and expressed in the membrane of the infected cell, then we start to see macrophages coming along and noticing those viral antigens, which will then trigger the immune response system. So the thing to take home from this is that viruses are highly selective for the host cell. For example, HIV virus targets T helper cells. The viruses don't have all the enzymes necessary to function outside the host cells, so they must use the host cell DNA to do that. But what the virus does do is contain the genes for the enzymes that they need to allow for infection and enable RNA viruses to be reverse transcribed so that they can be inserted into the host DNA, or they have the coding in them to provide the mechanisms for new virus release from the host cell. And any one of those things can be targets for drug design in order to combat viral infections. So our own cells do make an antiviral protein, and they do this in response to the production of interferon. And interferon is made by any cell that's been infected with a virus. So you can see here the first cell here with the new virus inside the cell starts to produce interferon. That cell will release interferon that is then received by neighbouring cells. And so you can see the little green dots binding onto the receptors of the neighbouring cell. And what that does is trigger the neighbouring cell to make antiviral proteins. And that means that we have this viral interferon telling all these other cells to make this antiviral protein in order to block the viral nucleic acid synthesis to make new virus. So our body system can do this, and through this chemical that the cells make called interferon. And we have learnt to make synthetic interferon so that we can give it to patients who we know have a chronic viral infection in order to prevent them from becoming ill and presenting with symptoms. So interferon's one of those drugs they might give to somebody who has a HIV infection in order that it doesn't become AIDS. So we looked at bacteria in part 1. And we now need to look at the differences between bacteria and viruses so that we can understand something about the differences in what we need to consider in antiviral drugs and why we can't use an antibacterial to treat a virus. On the right we have a diagram that shows an Escherichia coli and the size of it in comparison to a whole series of different viruses. And you can see that the bacteria, the Escherichia coli, the E. coli, is so much larger than any of those virus particles. But there's another thing that I want you to notice in that diagram called a phage, so a bacteriophage. And these are viruses that actually infect bacteria. And there are a couple of studies going on at the moment that are using these viruses that infect bacteria in order to kill the bacterial infection as an alternative to using antimicrobial drugs. So that's something to keep an eye on in the future. We might see that happening more and more often to combat problems with bacterial resistance. The differences between the virus and the bacteria. Bacteria have a cell structure. They are prokaryotic, so they don't have little mini organelles inside them. We can see them under the microscope. They can survive without invading a host cell, except for the groups called the rickettsia and the mycoplasmas. We can test the efficacy of a drug on a bacterial colony on an agar plate. So we can do antibiotic sensitivity tests to know which antibiotic to give to somebody specific for the bacteria they have. And that's a very useful tool. We can also gram stain them, which means we can work out which groups of bacteria are more, which group of antimicrobials are likely to work on any particular group of bacteria. Now that's very different to a virus. Virus is too small to see under the microscope. There is no cell structure. There is either DNA or RNA, but not both. There are no physiological functions going on in a virus when it's not part of a host cell. So it actually needs a living cell to be able to test a drug on. So we need to infect a living cell with the virus and then see if the drug can prevent the virus replicating or prevent the virus from releasing from that cell. And so they might use something like eggs in order to turn around and test drug sensitivity. So it's much more complicated process and a much more restricted process in being able to test a drug against a virus than it is to test a drug against a bacteria. So these flowcharts side by side, the blue one on the left is all about bacteria and how bacteria invade the cells and then how the immune system responds to it. And then the purple one is all about, or the violet one, mauve one, is all about how viruses infect the cells and then how the immune system responds to it. And you can see they're different. In the bacteria, we have the bacteria are phagocytosed by our macrophages or any of our other antigen presenting cells, and that means the antigen is then presented to our cytotoxic T cells and to our helper T cells. Our helper T cells go off and activate the B cells to make antibodies, and the cytotoxic T cells will destroy the bacteria through bacterial cell lysis. Now, antibodies that have been made by the B cells will tag onto any of the other bacteria in the system, or any of the antigens in the system, or the toxins that might have been released by the bacteria, and form an antigen-antibody complex, and that increases the likelihood of the T cells destroying them and increases the phagocytes, so these antigen-presenting cells and phagocytes, to actually move on and kill the other bacteria that are left in the system. Now, on the right hand side, looking at the virus, you can see that the virus needs to infect the tissue cells and uncoat itself and start using the cellular processes to make new viruses. Some cells may well be able to detect that a cell has been infected by a virus, and that then means that we get antigen presenting cells picking up the infected cell. And if they do that, then they present part of the antigen from the virus in the cell membrane, and that then activates T helper cells, and so we get B cells making antibodies, and that then helps all the cells that are infected by the viruses to be destroyed. The infection of the tissue cells itself causes, in some cases, the cells to present a piece of foreign protein in the cell membrane, which will also stimulate natural killer cells and also activate cytotoxic T cells, and that will help destruction of the cells that are infected with the virus. But notice, in every case here, we're destroying the cell that is infected by the virus. So we're destroying not just the virus, but we're destroying the host cell. Those infected tissue cells will also release interferons, and that will increase the defensive mechanisms of the neighbouring cells to avoid them picking up and ingesting the virus. Now, even looking at this, we can start to see that we have a slight problem if we're trying to test a drug against a virus, in that we can't test it on a person as such, for the simple reason that if we gave it to living human cells, then we know that the process of our immune response is to kill the cells that have been infected. So it becomes quite an ethical issue. So really it is a matter of doing it on something like egg cells, or if we can grow plates of living cells and test them on those, rather than testing them in actual people or animals for that matter. So, as I said, there are some real challenges around creating antiviral drugs, and consequently we have very few of them. Most of the clinical symptoms when a person is infected appear after the peak load of the virus has developed, so the illness progression is already determined by the virus. And the viruses can either be DNA or RNA coded, and they may have a protein coat, and they don't have any metabolic activity, so we can't kill them before they enter the cell if we're actually trying to get rid of them. So most of the drugs that we know will prevent viruses from replicating inside the host cells will actually damage the host cells anyway, so the host cells are lost. So as I said on the previous slide, even our immune system will turn around and destroy a cell that has within it a virus. So the host cells get destroyed in the process. So the few antivirals we do have are grouped as either the antiviral non-retroviral group or the antiretroviral group. Now the antiretroviral group are the ones that are used in HIV infection, and so we're not going to spend much in the way of time on that. If you do BMS 292, you will spend quite a bit of time on immunology, and you'll spend some time looking at HIV. In the antiviral groups, we have a couple of different groups. There is a miscellaneous antiviral group, and they're used in respiratory syncytial virus. We also have the DNA polymerase inhibitors, and they're used with things like herpes simplex virus 1 and 2, varicocele zoster virus, which is the one that causes chickenpox, and cytomegalovirus. Then there are the neuraminidase inhibitors for influenza A and B strains, and so they prevent the virus being released from the cells once it's been made. Now, we do have a number of vaccinations for the virus infections because we can use a portion of the virus to trigger a immune response by the body to make antibodies against that particular disease. And by using a portion of the virus, then we avoid actually causing somebody to be infected with the virus, but we can make the immune system respond to a portion of it. And so we have vaccinations for measles and mumps, rubella, chickenpox, human papillomavirus, influenza, and hepatitis viruses. So we've already noted that influenza can be a cause of pneumonia, and on the previous slide I mentioned neuraminidase, and so viral neuraminidase is an enzyme that when a virus has infected a cell and it started making new virus particles, this enzyme is needed to release those particles out so that they can go and infect other cells. So these neuraminidase inhibitors prevent that release of new virus to the cells, and in that way contains the infection so that our immune system can hopefully then go in and kill the cells that are showing some alterations in their membranes because of being infected by the virus. So the cytotoxic T cells and the natural killer cells can destroy these, and that way the illness doesn't progress and we don't see a peak load of virus developing. But in order to avoid that peak load of virus developing, these drugs need to be given within about 48 hours of the symptom onset. And so that can be fairly challenging. And we're talking about the symptom onset for the flu, not the pneumonia that can follow the flu with the same virus. So you'll notice that the drug names both end in MIVIR. So the VIR on the end is giving you some hint that they're antivirals. And we have two that are available to us for treating influenza. One is an oral inhalant, and it's quite well tolerated. The other is an oral dose drug, and it's not the preferred drug for those who already have something like bacterial pneumonia, or they have the flu if they also have underlying respiratory diseases or cardiac conditions. You'll notice that there's a list of quite adverse effects to the drug there. We've also noticed that respiratory syncytial virus is a cause of pneumonia in children, and for those that are less than 2 years old, they use a drug called ribavirin. Now, this particular drug inhibits intracellular mechanisms to stop new proteins and new RNA being produced by the virus. And again, it's an inhalation drug delivery, and the adverse reactions are not all that common. And being an inhalant is probably an easier thing with young children under 2 anyway. So that's fortunate. Now, before we move off the viruses, there is another foreign protein type particle that can cause infections that doesn't have anything to do with pneumonia, but we do need to know they exist. And these are the prions. They're infective particles. The only thing they consist of is a bit of protein. There's no nucleic acid to them, and they've been implicated in some of the neurodegenerative disorders. So these are things like spongiform encephalitis, which is Creutzfeldt-Jacobs disease, or the BSE, which is mad cow disease. So these particular, because they're proteins, that's all they are, is a piece of protein, they're particularly resistant to things like disinfectants and autoclaving. And there can be quite a delay from the time of infection to the time of presenting of symptoms. So let's look at the fungi. And the fungi structure is quite different from the bacterial fungi because there are organelles within the fungal structure. They do have a cell wall like a bacteria, and they do have a cell membrane like a bacteria. But whereas a bacteria has no organelles, a fungi does. And this means that they are termed eukaryotes. Now, human cells are also eukaryote structures. The difference being that our cells don't have cell walls. The term for a fungal infection is mycosis, and most of the fungal infections are in fact superficial infections, so they tend to be the skin, the lining of the mouth, the external genitalia. So athlete's foot is caused by a fungus, ringworm is caused by a fungus as much as it's called ringworm, and they're both variations on the fungus called tinea. And then the one we come across possibly the most in terms of things like nappy rash or overuse of corticosteroids, or every now and then we see it as a systemic infection in people who are immunocompromised, is Candida albicans. And so the disease is called candidiasis, and the cause is a fungus called Candida albicans. Now you can see here that the fungi comprise of the molds and the yeasts, and the yeasts grow as ovoid or spherical cells, whereas the molds are filamentous, so they're thread-like and quite multi-branching, and so you'll see the term hyphae being used. So systemic infections of fungi tend to occur in those who are already immunocompromised by something else. So either they're older, they've got some other infection in their system, they've got some other chronic illness that their immune system is very busy dealing with, and that leaves them open to being infected with a fungus as a systemic infection. And so pneumonia can be caused by Candida species, it can be caused by Aspergillus species, and also Pneumocystis jirovecii, which is the fungus that we see causing pneumonia in those who are HIV infected, where the HIV has moved on to AIDS. So how do we treat it? There are three groups of drugs, essentially, that we use in fungal infections. Flucytosine is an antimetabolite that stops the fungus from being able to make pyrimidine nucleic acids, and so it's used in any of the Candida species infections. There's a group of drugs called the azole antifungals, and you'll see azole in the end of their drug names, and these prevent production of a sterile compound, ergosterol, that is needed to make the fungal cell membrane. And so this can be used with some Candida infections, but also Aspergillus infections. The echinocandins, that you can see the drug names there end in FUNGIN, so fungin, which at least gives you a hint that they're working against fungi. They have significant activity against both those species, but they work by preventing glucan being made, which is needed to make the cell wall. So much like when we had the bacteria, we had drugs that worked at different points of the structure of the bacteria. We do the same thing here with the fungal infections. We have three different drug groups, and they work in three different ways to prevent the fungi from reproducing, either by stopping pyrimidine nucleic acids, or by stopping the cell membrane being made, or by stopping the cell wall being made. So here's a graphic describing that. So we have our fungal cell structure at the top of the screen in yellow and white there, And then we have our various drugs. So they've drawn out the cell wall, they've drawn out the cell membrane, and they've drawn out where the DNA is, and then they're giving you some of the drug examples to show where they work. Nystatin is one that used to see used a lot. And so, whenever I'm in class and I ask people if they've ever taken nystatin, they'll say yes. And invariably because they've had a thrush infection, so Candida albicans infection. And there's something to realize that if you take a broad spectrum antimicrobial, so something that's broad spectrum for bacteria, and because we have a population of normal flora in our system that are in competition for nutrients all the time, as soon as we turn around and take out some of the bacteria that are using up those nutrients, then the fungi get a chance to turn around and overgrow. And Candida species creating thrush, if you go to the GP and he says, here are your antibiotics, and then he's going to turn around to you and say, do you regularly get thrush whenever you're given a broad spectrum antibiotic? And that's when he'll say, well, if you do, come back and see me and I'll give you an antifungal. And often that drug will be nystatin or one of the azoles. So that overgrowth is because there are more nutrients available, and so the fungi can make use of the excessive nutrients that are resulting from the fact that the bacteria have been killed off in our normal flora population. Now, the parasites can be either the protozoans like an amoeba, or they can be the worms. So the technical word for a worm is a helminth, and if you look in any of the drug books they'll tell you whether a drug is an anti-helminthic, or the arthropods like scabies. So these are parasites that do a certain amount of damage when they invade our system, making use of the nutrients that our system provides for their survival, but at our expense. And so there's some pictures there to show you some examples of some of them. Worms are certainly multicellular organisms, whereas amoeba as a protozoan is a single-celled organism. So as multicellular organisms, these structures, the worms, actually cause physical injury through obstruction. So they take up physical space. But they also metabolize, so they use nutrients, they metabolize, they create waste products, and they can release toxic products, and that will damage the tissues around where they are and create an inflammatory response. Now, an infection from a worm is termed helminthiasis, and the really scary thing is that a third of the world is infected with worms. So a third of the world's people population has worms in their system of some description. Now, worms can cause pneumonia. We tend to think of them as being a gastrointestinal tract infection rather than something that ends up in our lungs, but they can end up in our lungs. It's not a common thing, but it can happen. Tapeworms on the bottom. The intermediate hosts of tapeworms are dogs and foxes. They form hydatid cysts in the liver, which then produce new worms. But about 20 to 30% of these cysts can actually form in the lungs, and they can remain asymptomatic for years until they turn around and decide that they're going to grow new worms, and so then create inflammatory responses in the lungs, creating that infection, lower respiratory tract infection. The flukes are ingested, usually because you've eaten them in another food. So most of the cases of fluke infestations occur in places like Southeast Asia and Central Africa or South America. And it's one of the reasons, some of our elders will not eat anything except well-cooked meat because of the fear of things in the food that are worms. And so they're the flukes. And then the roundworms. And the roundworms, it's a fecal-oral transmission, so you ingest them, they migrate to the liver from the gut via the porta hepatis, and then as the blood flow goes back out into the systemic circulation, it circulates through the heart, and as soon as it gets returned to the right side of the heart, it gets pumped into the lungs. And so we end up with the eggs being delivered to the lungs, which they then grow and form worms. So you can have worms as a cause of pneumonia. Now, most of the drugs we use to treat worms activate on worms that are in the gut. They're actually not well absorbed from the gut into the systemic system, and so that creates a bit of a problem in terms of treating them when they do move out of the gut. You'll notice again that this drug has a chemical similarity to the drugs that we used for the fungi, and consequently the name ends in AZOLES. If it's a drug that is going to kill the worm, they're termed vermicidal. If it's a drug that's going to kill the eggs of the worm, then they're called ovicidal. And what they do is actually block the glucose uptake by the worms and any other parasite that might be around to stop them being able to make cellular microtubules. Now, the microtubules form the structural elements within the cells that create the layout for where the ribosomes are and where the nucleus is, and in fact the spindle is created of microtubules for reproduction, and so producing microtubules is a key metabolic function of any living cell, including our own. And so these drugs block glucose uptake and damage the microtubules and the production of microtubules. So they're indicated for most of the worm species, and you might recognize some of the names here. If you've ever wormed a farm animal, you've probably seen ivermectin, and you've quite possibly used ivermectin on your pets and on your farm animals. Parental's another one, and parental actually works by depolarizing the action potential in nerves and muscles and causes the worm to be paralyzed, and that means it can be dislodged and excreted. Ivermectin does a similar thing with chloride channels, causing muscle paralysis, and praziquantel causes cell membranes to increase their permeability so that the cell loses intracellular calcium, again resulting in paralysis. So these three drugs here that don't belong to the azole group work through a mechanism of affecting the action potential or the muscle contraction so that the worm becomes paralyzed and can be dislodged. Now, the last one, praziquantel, is actually well absorbed from the gut, so it would be the one that you could use when these things move out of the gut. Now, the worms had a cellular structure that was much like our own, being a multicellular living organism, and so do the protozoas. No cell wall, but certainly a cell membrane, and in the case of protozoas, there may be a flagella to help with movement. Most of them are found in water habitats such as Giardia intestinalis. Some exist as parasites in other animals and insects, so an example of that is the malaria protozoan that lives in the saliva of the Anopheles mosquito. And some have a two-stage life cycle, so the example I've got here again is malaria because it's relatively easy to describe. And so there's a sexual cycle that occurs in the mosquito where there's male and female gametes produce sporozoites, and then the asexual cycle occurs in humans. So the sporozoites are delivered in the saliva of the mozzie when it bites you, and they migrate from the blood to the liver and develop these forms that then burst out of the liver cells as merozoites, and then the merozoites infect the red blood cells. So malaria has this two-stage sort of cycle and this two-stage of creating symptoms in the body. So if an amoeba can actually penetrate the gut mucosa, then we start to see movement away from the gut into places like the liver, occasionally the spleen, the lungs, or the brain. Certainly if they stay in the gut, they create diarrhea and abdominal pain, and anybody who's had amoebic dysentery will tell you it's pretty horrible. So it's called by an amoeba called Entamoeba histolytica, and it's a fecal oral transmission process in the water, and because it can migrate from the digestive tract into the liver, form a liver abscess, and that liver abscess can then start extending up through the diaphragm and into the lungs. And so we then see, or it can be transported through the blood flow or through the lymphatic systems into the lungs, causing pneumonia. So amoebic dysentery can actually lead to pneumonia. So again, when we look at the drug names for treating an amoebal or a protozoal infection, you can see here that metronidazole and tinidazole end in azole, which we've already come across for one of the drug groups to treat the worms, and we've also come across as one of the drug groups to treat the fungi. The first one, metronidazole, is a prodrug, and the infecting organism actually activates it to the active drug. It has both these drugs have some selective toxicity for anaerobic species, and they're quite well absorbed from oral administration, so they can be used across the board.